Welcome back everyone to part 2 of topic 9 on the digestive system. And here we're going to look at some of these accessory organs or glands, being the liver, the gallbladder, the pancreas, and then we're going to also talk about chemical digestion. Here are the learning objectives for this part of the topic. Be able to outline the anatomy of the liver, gallbladder and pancreas. Be able to compare and contrast the functions of the liver, the gallbladder and the pancreas. Describe particularly the anatomy and histology of the liver. And then outline the relationship between the liver, the gallbladder and the pancreas in terms of ducts. Be able to list and describe the functions of the liver and identify blood and bile flow through the liver. Be able to summarize the regulation of bile secretion and release, and list the pancreatic secretions and outline their function, and finally outline the production of bicarbonate in the pancreas. We'll start with the liver, and the liver, as you can see here, is the largest internal organ in the body, and it weighs sort of on average about 1.4 kilograms. It's located in the right upper quadrant of the abdomen, and it kind of lies against the diaphragm there. It consists of two major lobes, the left and right. You can see the right lobe there and the left lobe there, and also two minor lobes, and they are the caudate lobe and also the quadrate lobe. The porta is located on the inferior surface, and this is the location where vessels, ducts and nerves exit and enter the liver. Remember, the word porta kind of means door in Latin. The hepatic portal vein, the hepatic artery and the hepatic nerve plexus all enter the liver at the porta, and the lymphatic vessels and the two hepatic ducts exit. So in terms of these hepatic ducts, we have the right hepatic duct and the left hepatic duct, and they unite to form this common hepatic duct. The common hepatic duct then unites with the cystic duct, which you can see here from the gallbladder, to form the common bile duct. So wherever you see a word beginning with hep, you know that we're talking about the liver. Here, hepatic ducts, but you could be talking about inflammation of the liver being hepatitis. So in a little bit more detail, looking at the relationship between the liver, the gallbladder and the pancreas in terms of these ducts or tubing systems. If we start here in one, right, the hepatic ducts, they're carrying bile from the liver lobes, and they combine to form that common hepatic duct, okay? The common hepatic duct combines with that cystic duct that I spoke about on the previous slide from the gallbladder to form the common bile duct down here. The common bile duct and the pancreatic duct then combine to form what is known as the hepatopancreatic ampulla, which you can see there. Yeah. And the hepatopancreatic ampulla empties bile and pancreatic secretions, which we're about to learn about in this recording later on. So things like pancreatic enzymes and bicarbonate into the duodenum at the major duodenal papilla, which is this sort of raised bit here. And then the accessory pancreatic duct empties pancreatic secretions into the duodenum at the minor duodenal papilla when that is present. Now going to look at a little bit of liver histology. So what you're looking at here is a lobule. Okay, so connective tissue septa branch from the porta into the interior of the liver, and that divides the liver into these lobules. Nerves and vessels and ducts all follow the septa. Within each lobule, then, there is a portal triad at each corner, okay? And within the portal triad, there's three vessels. The hepatic portal vein that is carrying, that nutrient-rich deoxygenated blood. We also have a branch of the hepatic artery, which is carrying oxygenated blood. And then we have the hepatic duct, which is carrying bile. In this, we also have this central vein then in the center of the lobule. And all of these central veins from all of the lobules then unite eventually to form the hepatic veins that exit the liver and empty into the inferior vena cava. Now, just some further information about the histology. We have these hepatic cords. So these radiate out from the central vein, and they're composed of hepatocytes, which are these liver cells. Hepatic sinusoids then, are those capillaries that are very leaky, and they're found between the cords, and they're lined with endothelial cells. And also this particular type of macrophage, it's called a Kupffer cell. So these are these liver phagocytic cells or macrophages. And we also have these bile canaliculus, which kind of means little canal. So these little canals that are, collecting the bile that's made by the hepatocytes, and they're located between the cells within these cords. Now, hepatocytes or liver cells have a diverse range of functions. They make bile, okay? They're involved in storage. Don't worry, I'm going to get onto all of these on the next slide. They're involved in the interconversion of nutrients. They're involved in detoxification. And I guess when we think about the liver, we always think about it detoxifying different things. That might be things like, drugs that you might have taken for, medications, and also foods, and also alcohol, and lots of things that have to be detoxified. Remember, I talked about ammonia in the urinary topic, and that's converted to a less toxic urea in the liver. Okay, I'm getting ahead of myself. Phagocytosis of any debris and synthesis of blood components. So let's go through all of those things on the next slide. So the first function then is bile production, and the liver does make about 600 mils to a litre of bile per day. Now, it contains bile salts. It also contains bilirubin, remember the breakdown product from red blood cells. It also contains cholesterol, and it also contains fats and some of the fat-soluble hormones, vitamin A, D, E, and K, and also lecithin as well. Now, lecithin is a mixture of fats, and it's kind of essential to the body. It's used, it's converted to choline, and that's used then to make acetylcholine, the neurotransmitter, and it's also used to move fats. Now, what is the purpose of bile production? Well, it neutralizes and dilutes stomach acid. So the chyme coming from the stomach has the pH of it is just way too low for the pancreatic enzymes to function. So the bile neutralizes it and dilutes that stomach acid. Bile salts also emulsify fats, and most of the bile salts then are reabsorbed in the ileum and recycled. Okay, what is emulsification? Well, it's kind of the first step in the in lipid digestion, and it's the transformation of large lipid droplets into much smaller lipid droplets, because the enzymes, that catabolize sort of lipids are water soluble, so they can only kind of work at the surface of the droplet. So, by making these larger droplets it turn into much smaller droplets, that's going to increase then the surface area for those pancreatic lipases then to work. Now, secretin, that hormone that's secreted from duodenal cells, stimulates bile secretions, increasing the water and bicarbonate ion content of the bile. The second function then is storage. So the liver does function as a storage site for lots of different things. Glycogen. So excess, glucose enters hepatocytes and it's converted to glycogen. Fat, the fat-soluble vitamins, as I mentioned, A, D, E, and K, copper and iron. So they're all stored in the liver. And remember, it's that hepatoportal vein that's, bringing blood to the liver that's really rich in all of these nutrients. So these hepatocytes are playing a really important role in regulating blood glucose levels, because again, just going back to this hepatic portal vein, it's bringing all of that glucose to the liver, where the hepatocytes can store it and then release that glucose when needed. The next function then is nutrient interconversion. Now, very often, our diets, they're not sort of that well balanced to produce, enough sort of nutrients to supply what our body cells need. So say, for example, you might be on a really high protein diet, and therefore you might not have enough lipid or carbohydrate in your diet. So this is where the hepatocytes come in, because they can interconvert those amino acids, in this case, to energy producing compounds, such as ATP, lipids, and glucose. Another example of nutrient interconversion then is the hydroxylation of vitamin D. Remember that first step of vitamin D synthesis occurs via UV exposure of the skin. That travels to the liver, where hydroxylation of it occurs, and then that form then travels to the kidneys, where it is hydroxylated again to its active vitamin D form or calcitriol. Detoxification. Yeah, as I mentioned, the hepatocytes, they're very busy cells in terms of detoxification. The example we've given you there is to remove, ammonia and convert it to urea. And that urea, of course, then is eliminated in urine in the kidneys. Other sort of detoxification pathways might, form a compound or a metabolite or something that then gets excreted in bile and ends up in the feces. Phagocytosis then is carried out via these Kupffer cells, and these Kupffer cells line the sinusoidal or sinusoid wall of the liver, and they're really busy as well. They phagocytize worn out and dying red and white blood cells, also any bacteria, and any other debris that they can engulf. And finally, the liver is so important, everybody, for making all of these proteins that are really valuable to the body. So the albumins, remember the plasma albumin, that it forms most of the osmotic pressure of the plasma. Fibrinogen, that's converted to fibrin in the clotting mechanism. The globulins, so the immunoglobulins, so think of, antibodies. Heparin, and also those clotting factors that then feed in to that clotting cascade, which is a part of hemostasis as well. And the final thing we're going to look at here is just a very simple schematic looking at blood and bile flow through the liver. If you start over here on the right hand side, we have the hepatic artery, which is carrying that oxygen rich blood to the liver. At the same time, we have the hepatic portal vein, which is bringing deoxygenated but nutrient rich blood from the digestive tract to the liver. We've also got the bile duct here. So that bile that's made in the liver, the bile duct then brings that liver back to the duodenum of the digestive tract. And then over here, we have the hepatic vein that's carrying that oxygen poor blood out of the liver and then merging with the inferior vena cava, which will then take that deoxygenated blood back to the right side of the heart. So the gallbladder is a sac-like structure, and it's located on the inferior surface of the liver. It's about 8 centimeters long and it's 4 centimeters wide. And it's lined with mucosa that are folded into rugae, and those rugae folds can sort of, disappear as it fills up with bile. It has an inner muscularis layer and an outer serosal layer. Bile is continuously being produced in the liver, and it's transported to the gallbladder where it is stored, and it can store about 70 mils of bile at a given time. And the bile is also concentrated in the gallbladder, by removing water and electrolytes, and that then leads to the concentration of the bile salts and the pigments. The gallbladder is stimulated to contract via cholecystokinin, which is a hormone that's secreted from intestinal cells. And it's, under vagal stimulation via the parasympathetic nervous system. The bile then exits through the cystic duct and then onto and into the common bile duct. Now, I'm sure you've all heard of a clinical condition called gallstones, which is due to precipitated cholesterol. You're looking at a gallbladder here stuffed with these gallstones. All right, so remember, the cholesterol is not water soluble, and it's usually the bile salts that keeps it in solution. However, these gallstones can occur due when the cholesterol precipitates out of solution, and that might be due to excess cholesterol in the diet, or it might be due to the gallbladder for some reason, concentrating the bile too much. Occasionally, one of these gallstones can slip out and can block the cystic duct, and when that happens, that's going to block the flow of bile and therefore interfere with lipid digestion, so that's going to have to be surgically removed. Other times, these stones can, go further down and block the pancreatic duct, and that's much more serious because it's going to end up causing pancreatitis. And many people, particularly females, end up having their gallbladder removed. And these gallstones can occur because of drastic dieting, and that's kind of thought to be caused by a change in the ratio of the cholesterol to the bile salt. The last thing we're going to say about bile is the control of bile secretion and release. So bile, including bile salts, is produced by the liver and it's stored in the gallbladder. Okay, the bile salts are reabsorbed in the ileum and stimulate more production of bile, so it's recycled, right? The hormone secretion from the duodenum also stimulates bile production, and the cholecystokinin then stimulates the release of bile from the gallbladder. We're now going to talk about the pancreas. And remember that the pancreas has both endocrine function, so producing those hormones, secreting particularly insulin and glucagon that regulate blood glucose levels, but it also has an exocrine function, and that's what we're interested in here. If you look at the pancreas here, you can see that it has a head region, a body, and a tail. Its endocrine function is due to the presence of these pancreatic islets that secrete those hormones, so the alpha cells secreting glucagon and the beta cells secreting insulin, and also there's somatostatin as well that regulate both insulin and glucagon. But these exocrine functions are due to these groups of acini, which are these kind of grape-like clusters or berry-like clusters from lobules that are separated by these connective tissue septa. Now, intercalated ducts lead to intralobular ducts, leading to interlobular ducts, which then drain into the pancreatic duct, which you can see here. So all of those digestive juices enter this pancreatic duct. Remember again that the pancreatic duct joins the common bile duct and then tips into the duodenum at the hepatopancreatic ampulla, which is controlled by the hepatopancreatic ampular sphincter. So what's actually in this pancreatic secretion? Well, firstly, we have an aqueous component, and that's produced by the columnar epithelium lining those smaller ducts. Sodium ions, potassium ions, bicarbonate, and water. Now, the bicarbonate is obviously acting, as a buffer, and it increases the pH by inhibiting pepsin and providing proper pH for the pancreatic enzymes. The enzymatic portion then, there's lots of different things in there, as you can see, and a lot of them have either ogen at the end of them or pro at the beginning. And that means they're in an inactive form of a particular substance. So we have trypsinogen, we have chymotrypsinogen, we have procarboxypeptidase, we have pancreatic amylase, we have the pancreatic lipases, And we also have these other enzymes called deoxyribonucleases and ribonucleases. So you can see that these are either straightforward enzymes or they are precursors or inactive forms of particular enzymes. So what actually happens in the duodenum in terms of all of this, activity of these pancreatic enzymes? It's due to an interaction of duodenal enzymes and also those pancreatic enzymes. We have this enzyme called enterokinase, which is attached to that microscopic brush border of the duodenal mucosa. And this enzyme activates trypsinogen. So it converts trypsinogen to trypsin. And we know that trypsin is a proteolytic enzyme. So it's going to split proteins and continue the digestion of proteins. But trypsin also activates chymotrypsinogen to chymotrypsin. And trypsin also activates that procarboxypeptidase to carboxypeptidase. So, as I mentioned, trypsin is proteolytic. It's an enzyme that splits proteins, but so is chymotrypsin, and so is carboxypeptidase. So they're going to continue the digestion of proteins, from the peptides down to the tripeptide, dipeptide, and single amino acid level. Pancreatic amylase continues the digestion of starch. Pancreatic lipase digests lipids, and the deoxyribonucleases and ribonucleases digest RNA or DNA and RNA respectively. Now this slide is just sort of describing bicarbonate anion production in the pancreas. You don't need to memorize all of this, but it will look quite familiar to you in terms of, water combining with carbon dioxide and that carbonic acid and all of that. But we do have the elements of the blood vessels. We also have these intercalated duct cells that are producing the aqueous component of the pancreatic juice. And then you can see here that the bicarbonate anion then is transported into the duct, and it's going off then to the interlobular duct. So it's produced from carbon dioxide and then transported. Okay, everybody, that's all we're going to say or talk about for the liver, the gallbladder, and the pancreas.